And look, are you okay with this microphone? Maybe it's just today it's working a little bit better, perhaps. Okay. All right. Thanks so much.
Thank you. So, thank you so much, Anna, for the beautiful talk. And I'm a little bit cross with you, John, for putting me after Anna's very tough facts to follow, and I'm not sure that I'm up to the task. So that is my disclaimer. I'm joking, obviously. Today I'm going to be talking about alternative means of extracting some relevant information as they relate to some stimulus and some dimension of the stimulus of interest, other than just looking at univariate amplitude responses across layers. This is not to say that univariate amplitude responses and looking at amplitude differences is not meaningful. I'm just saying, I just want you to be aware that there are different ways of looking at your data. So just a very brief outline. I'm going to show a little bit about multivoxel pattern analysis, specifically decoding. I'm talking about decoding. In this case, will be linear SVM primarily. I'm going to talk a little bit about, again, spatial resolution of univariate, and we already covered this in previous lectures, so I'll be quick. And then we're going to see if we can maybe estimate the spatial precision of multivariate decoding. Then we're going to talk about representational similarity analyses, and then finish with some encoding models, some simple GLM models, and some more advanced population receptive field mapping. And I'm going to provide a little bit of background and then some applications to laminar fMRI of all of these methods. A little bit of background. So this is one of the papers that really inspired me very much and got me interested in the field of multivariate analysis. The paper by Kamitani and Tong 2005, where they showed that even a 3T with voxels as big as 3 millimeters isotropic, they could decode using a linear support vector machine orientation tuning in B1. This paper sparked a big debate in the field that is still going on right now as to what are the information, how is it even possible to decode information that are so well beyond the nominal resolution of the voxels. The original explanation was that when you pull all these voxels together, you can retrieve some local biases in orientation preferences, in this case, arising, for example, from variability in vasculature or neuronal distribution. What I wanted to do? No. From then, there's been a lot of interest in multivariate analyses. And there's this really nice paper that I like from Nico Krieger-Score 2007, where he talks about analyzing information versus analyzing activation in fMRI. And what he means by that, when he talks about activation, he's talking about the univariate amplitude, so within a given ROI, let's say. So for example, this red circle here. Can you see my person? This red circle here, average all the voxels together and look at the mean amplitude. And then that's the activation analysis. And then instead, the information analysis would be, instead of like averaging these voxels, you don't do any smoothing, and you actually exploit the pattern of information contained in the voxel and see whether that carries any meaningful information. And his example that he provides in his paper that I very much like is that you do have two different conditions which have a specific neuronal activity pattern. And sure, this information is blurred, distorted, even displaced by the sluggish hemodynamics response, but it's still present in voxel population to a degree that allows to differentiate, for example, neural representation, in this case, between these two phases. So very briefly, this is probably a redundant slide for most of you, but how does a linear support vector machine work? So let's imagine we have two conditions, condition A and B, and we want to know if a given region of interest carries different information about these two conditions. So we can assume that we've got 3 trials for condition A and 3 trials for condition B. And the way SVM works is it replots in a way you can think your amplitudes in a multidimensional space whose dimensions are the voxels. So here's an example on a very simple 2-dimensional space where we replot the activity elicited by condition A and B, all the trials condition A and B, in this new space where my dimensions are voxel 1 and voxel 2. And then we can use a portion of our data to train our SVM to optimize the distance between these points, and you do so by optimizing a set of weights that allows you to stretch the space, this new multidimensional space, to maximize this distance, and then you compute these support vector here shown as these dotted line. And then what you do on an unseen portion of the data, you see how well these weights and support vector generalize to other portions of the data. You count the instances in which you are correctly classifying your labels, and that gives you information as to the decoding accuracy of that region. If you can successfully decode above chance this information, then you conclude this region carries some information about some aspect or dimension of the stimulus at hand. You do this as a cross-validation in a cross-validation manner. So you split your data in testing and training folds. And the way you split your data and normalize your data is actually very important, but I'm not going to go through this right now. If you want, we can discuss this later. So one example of MVPA decoding, in this specific case, linear SPM, was in our paper in 2015 with Lars Muckli in current biology. And so we built on the paradigm that Lars and Fraser Smith developed in 2010 in PNS, where they were asking questions whether it's possible to decode feedback contextual information in occluded areas. So what they did was presenting different images like the ones shown up here, either in a feed-forward condition, so fully stimulated, or in a feedback condition with the bottom right-hand quadrant occluded with a white occluder. They mapped out the retinotopic representation of the occluded quadrant in V1 and confined subsequent analysis in these regions, and they demonstrated they could successfully decode information even in the white patches, and concluded that this must be some filling in effect coming from higher level areas of feedback. So we repeated this experiment at 70 with 0.8 isotropic voxel. This is like getting echo data. And we looked at V1, V2, V3. I'm just going to show results from V1 today. Again, we had like in this in-session localizer with this flickering ratings to determine the retinotopic representation of the bottom right-hand quadrant and confined subsequent analysis into this region. Performed layer cortical depth segmentation. In this case was using Brain Voyager, and we have 6 depths. And then we looked at the decoding accuracy across depths for our feed-forward, so for our visually stimulated portion, and our feedback, so our occluded portion. And there are some results. So what we saw is that oh, I got it. All right. So what we saw is that we could, unsurprisingly, successfully decode information with these 3 highly different images in V1 with high accuracy across all layers or cortical depths for the feed-forward, they are shown in red, where we're speaking in the middle. And then for the feedback, we were only able to, so for the occluded portion of the experiment, we were only able to decode successfully above chance information about the stimuli in the outer layers. So this led us to conclude that feedback contextual information arrives in the outer layers in V1. This one? Yeah? I don't know. I can tap my chest if you want, but there's no mic here. An interesting aspect of these results is that if you look at the univariate difference in this specific case, we didn't observe any differences between the 3 different images. Here is plotting the univariate differences for the 3 images in 3 different colors across the depths in V1 for the feed-forward and the feedback condition. And you can see that maybe in the feed-forward there may be some amplitude differences with the green bar being a little bit larger than the other 2 in the middle layers, but the error bars are very, very large and there was nothing significant there. This is not very surprising because even though the images were very different in amongst each other, low-level visual properties were equated, and once you average all the voxels within the retinotopic representation, you really lose these structural differences across images. So in this case, linear SPM was really useful in decoding information that would have otherwise gone amiss by looking at univariate amplitudes. Can we do a little bit more in-depth analysis of layer information using MVPA, and specifically, can we look at layer communication profiles across cortical depth? So this is what we did in the same paper using Brain Voyager. Again, we unfolded each single layer into grids of same dimensions using Laplacian equivolume, in this case, grids. I'm not going to go into the detail how this was achieved. You can ask Farouk, who's probably involved in the designing of this algorithm in the first place. And by doing this, now we are in a position where we have the same number of pixels across all layers. So what we can do is we can compute our retinotopic representation of the target in one layer, here shown in white. So and then we can project this across the other layers. And this then allows to have the same number of voxels across layers and train our SVM on one layer and see how well it generalizes within layer, but also across layers. And we can repeat this trick obviously for all layers. So we did it for the innermost layer, I showed you before, and this is an example from the outermost layer, where we compute this logical on the outermost layer, or masking the outermost layer, and then project it down across layers. And so these are the results between feedback and feed-forward condition. In the feedback, we saw that this layer communication occurred primarily in the outer layers, the two outermost layers, and was not significant anywhere else. In the feed-forward condition, we see not only that the diagonal is larger than the off-diagonal, which is a good sanity check, because that means that when you stay within the layers, you're decoding your feed-forward information better. But you also see there's a peak of like this distribution of information up in the middle layers. This also maybe tells us something about the precision of our multivariate pattern analysis. You can think this almost of like as a two-dimensional point spread if you want. But yeah, so that was our conclusion, and that was our hack to try and look at the layer communication profile. But what if we don't have a specific hypothesis or a specific region of interest that we're interested in and we want to have a more exploratory analysis? Can we still use a linear support vector machine or multivariate decoding in general? And the answer is yes. So again, another paper by Nico Kriges-Korte in 2008, where he put forward this idea of this multivariate searchlight. The idea is rather simple. You have a sphere of a given diameter. This, in this case, is like 4 millimeters, a voxelated sphere, I should say. And then you center the sphere on a given voxel, and you use that as your region of interest. You perform your SVM decoding, and then you start sliding the sphere across the brain, centering all neighboring voxels, and keep repeating your SVM decoding. So what this leads to is a map of decoding. Obviously, it's a blurred map, depending, because obviously you're kind of like, you know, subsampling in a way your space. But it can allow you to look at decoding performances across the brain. So we did this with layer fMRI, so 0.8 data. This is the work of Logan Dowdle, one of my postdocs. He used, in this case, FreeSurfer. He acquired 0.8 data with face task. He asked participants to do either a gender task or a stimulus-relevant color task, and then asked whether he could decode differences between the two tasks across the ventrotemporal cortex. This lab was quite large and allowed for looking at multiple areas at the same time. As I said, he computed cortical depth segmentation using FreeSurfer. He divided the cortex into 12 bins, and then he grouped into 3, so inner, middle, and outer depths. And then he used the searchlight approach with a searchlight of 4 millimeters on an inflated surface. And here are some results in the inner, middle, and outer depths. These are highly significant, threshold of 70% accuracy. You can see there's lots of spots around the brain, and I think this is very, very promising. I'm going to focus on one result that seems to be replicable across subject. But again, these are very much pilot data. And this result is looking at the fusiform gyrus and maybe FFA, maybe not FFA. But what we observe is that decoding accuracy was most pronounced in the inner depth of the anterior portion of the FFA, as well as the middle depths of the anterior portion of the FFA. But as you approach the outer depths, then it was the posterior portion of the FFA that was showing highest decoding accuracy. The other interesting aspect of this study, which actually touches on to what John was saying about determining the size of the ROI, which is really important, is that we had an online localizer, face localizer, and by online I mean in session, I wanted to say, not online, face localizer to identify FFA and OFAs. And if you look, that's a bit unfortunate because the rotation of the brain is slightly different, so it's not super easy to see. But if you look at the patches of voxels that are important for decoding in this case, they actually fall outside of the region of interest. This part here and this part here would be surrounding here and here, And if you look at the anterior portion as well, is like surrounding the center of mass of what we identified to be the FFA. And he carried out the same decoding analysis within the region of interest as determined by our phase localizer. And actually, the decoding accuracy was below chance. So using searchlights, for example, allows you to look at areas outside the specific region of interest, and it can be a more like, data-driven approach. So MVPA, such as, for example, linear SVM, allows to analyze the information contained in a multivoxel pattern of activity. And there are cases in which it can be useful in retrieving differences that otherwise can go amiss with standard univariate analysis. But does this imply that MVPA possesses a higher spatial precision than univariate amplitude analysis? Is it sensitive to fine-grained structures in the pattern, in the multivoxel pattern of activity? Or what we see is just the results of a clever averaging. And by clever averaging, I mean enhancing the weights of meaningful voxels that carries the information about the dimensions of the stimulus at hand, while weighing down voxels that do not impact classification accuracy. So to this, I'm not going to go into details about this, but I just wanted to mention there's like a big debate still ongoing about what are the information that MVPA is relying on to decode, for example, orientation coding, whether a coarse map or more finer maps. And there's this idea of radial bias, which could explain at least part of what we see in the orientation decoding. I just wanted to make you aware that there's debate out there. We're not going to go into that debate, but it's good to know that people are thinking about this. So to determine, to try and understand a little bit what is the spatial precision of our MVPA and whether that is actually higher than the precision that we can achieve with univariate amplitude, we need to think about how we can quantify the spatial precision. In a univariate perspective, it's relatively simple, especially if we are exploiting the written properties of V1, like I showed you the last couple of days. We can compute functional point spread function, and just as a very quick recap, this is how this is done on an inflated cortical surface. We have two retinotopic stimuli, a target and a surround stimulus, compute the linear contrast between these two to determine the empirical medial-most edge of the target stimulus, and then we can look at the activation elicited by the target stimulus only, superimpose this empirically derived edge, draw some traces that are perpendicular to this edge, and then quantify how fast or how sharply the activation ceases as a function of distance from this edge. And to quantify this, then we can fit our point spread model, which is a stepwise function representing infinite spatial precision, convolved with a Gaussian point spread, the full width half max of which is one of the parameters we want to estimate, the most important parameter we want to estimate, which is the parameter that's going to tell us what the spatial precision of our data is. And so this is what we did in our 2021 paper, and we saw that for 0.8 grain echo data, our estimated spatial precision 1.4 millimeters isotropic, which is very much consistent with what Dennis Chemos showed in 2017, between 0.99 and 102 for grain echo data. So this is maybe a little bit of a problem, because we are saying, well, using MVPA, we can retrieve information that I'm not going to say goes beyond the nominal resolution of the voxels, but at least is as precise as the voxel resolution. And here we can see that at univariate level, our spatial precision instead is not, it's blurrier than the voxel resolution, as we know that. So can we try and quantify this spatial precision for our multivariate decoding? So one data-driven way that we developed to try and get to this point was based on the artificial misalignment of the training ROI relative to the testing ROI, the other way around, the testing ROI relative to the training ROI. It's quite a simple trick. So you can think of your region of interest here, shown as a gray ROI as a cartoon representation here, where we want to carry out an analysis. We train our SBM on this region of interest and test it on the same region of interest, and then we test decoding performance on a shifted, artificially shifted region of interest, in this case to the right, 1 voxel, 2 voxel, 3 voxel, 4, 5, and we repeat this shift in all possible dimensions. And then we're going to assess how this artificial misalignment impacts decoding accuracy, with the idea that if we have fine-grained information, as little as a voxel shift should lead to a significant decrease in decoding accuracy. So before showing you some data, to test this, we did some very simple simulation with some very naive synthetic data. We started from original 0.8 dataset to estimate mean and standard deviation of the data, and then we reconstructed a high-resolution pattern with the same descriptive statistics. We injected some white noise to simulate variability across different trials, and then we convolved this pattern with Gaussian kernel of either 1, 2, 3, or 4 voxels to simulate different spatial precision of the patterns. And then we went on to perform our misalignment approach on this control dataset, synthetic dataset. And what we observed is that without any voxels moving, one voxel shift completely destroys the SVM decoding accuracy, returning back to baseline. If you do one voxel smoothing, then you need one more shift and so forth. 2 voxel need 3 shift, 3 voxel and 4 shift and so forth. So this validation kind of reassured us that maybe what we were doing could have been a viable option to determine what's happening and the spatial precision of MVPA. So here's an example with the feed-forward data from the Codon Biology paper across the different layers, and you can see that we have inner to outer layers here, inner to outer cortical depths, and then within each depth we can see the decoding accuracy for the original ROI, and then for the ROI shifted 1, 2, 3, 4, and 5 voxels. And what we observed is that as little as 1 voxel shift. Oh, and we repeated this analysis with different type of decoding algorithms, in this case, linear discriminant analysis and naive Bayesian classifier, and we achieved comparable results. What we observed is as little as 1 voxel shift significantly impaired decoding accuracy. However, unlike we saw in our simulation data where everything is super well behaved, obviously, and we did not put a lot of effort into try and simulate a realistic spatial and temporal autocorrelation of fMRI data. But unlike our simulation, we see that while 1 voxel shift significantly impaired decoding accuracy, there's still a lot of information contained in this misaligned ROI. We can get up to 60 or even 70% our decoding accuracy. So maybe this is indicating that this multi-voxel pattern of activation contains both some fine-grained information about the stimulus at hand, but also has some coarser information. So how does this relate to the question that we were asking? Does this imply that MVPA possesses higher spatial precision than univariate, and whether this is sensitive to fine-grained patterns or is simply an outcome of this clever statistical averaging? And I don't think that these two hypotheses are necessarily mutually exclusive. I would be very happy to find out that, we don't have an answer for that, but I would be very happy to find out that the reason why we can exploit this fine-grained structure is because of the stretching and these weights that we apply to our multidimensional space. All right, next I'm going to talk about representational similarity analysis, which is another way of looking at this neural representation and to exploit the multivariate or multi-voxel pattern of activity. So that's a relatively simple implementation. You can think of here, we got 2 runs with 2 conditions, bottles and shoes, 3 trials per condition, and then we've got a given number of voxels. So run 1 would be a 6 by n matrix, where n are my voxel, obviously 6 are my trials. And then you can perform a simple correlation with this, which then would lead to a 6 by 6 matrix. And the correlational values, you can do the similarity or dissimilarity. Dissimilarity would be 1 minus r, or similarity just like Pearson's r. And the correlational values will tell you something about how similar these neural representations are. You can do this in a split half way, or you can do it within each run. If you do split half, the diagonal is going to be a meaningful diagonal, but the matrix is going to be asymmetric. And the reason why I mentioned that is because to assess the geometry properties of representation similarity analysis, what we can do is multidimensional scaling, which is pretty much based on PCA. What we do is is a dimensional reductionality technique where we perform pretty much PCA on our data, and we get we replot our data on a new multidimensional space, in this case again a 2-dimensional space, according to the eigenvector that explain most variance in our data. To do this though, you need a symmetric diagonal, a symmetric matrix with ones or zero over the diagonals. And so you need to either like average top and bottom if you're doing split alpha, or you need to look at within run correlations. Here's an example of Nico's data from 2008 in the Neuron paper. These are human data where he showed a lot of different stimuli to subjects in the scanner, and these stimuli can be broadly subcategorized in animate and inanimate, and within the animate you've got faces and bodies, and human faces and non-human faces. And the cool thing about representation similarity analysis that I like very much is that it allows you to test competing conceptual models. So for example, here we can see two different models, an animate-inanimate model, which is just like basically highly correlated within the animate and highly decorrelated when you're looking at animate and inanimate, or a face-non-face model where high correlation is determined by whether you're looking at a face or not. And then you can do a simple Spearman correlation between your data and your model. And here we got many more models. That's like from Nico's paper, I think it's 2008 as well in Frontiers in neuroscience. We have many competing models, and we can see how well our model, our data correlate with our model. And then basically using a winner-take-all approach, we can make assumptions as to the neural representation and the organization of the neural representation in the region of interest. So we used RSA across cortical depths in Ished's paper, was published in 2020 in the Journal of Neuroscience. This paper was based on the face localizer from Analytical Spectors lab that holds several categories, specifically 4 categories. I don't know if you can read this. We've got characters, bodies, objects, and places, and within each category, you've got 2 exemplars for each category. And this allowed you to have 3 competing models: a domain level model, whereby the correlation would be very high, so the neural representation would be very similar within categories. You got a category level model where the neural representation within domains, and the category level model with the neural representation would be very similar within category, and then you got a cortical depth model with related representation geometry down here. These are obviously simulations. So here are some data. Ished looked at lateral and medial VTC, and he compared this obviously data with the model. And while he didn't find meaningful correlation between the depth level model and the data, and in fact the domain level model was the one that was explained the best, he did observe some interesting cortical depth differences, where the dissimilarities of the domain level, within domain level representations were most pronounced in the inner depth compared to the outer depths. This could be feedback in the inner depth. It could also be just related to blurring, related to large draining vessel present in the PL surface, which would lead to comparable results. So SBM is very useful, and I like using it. However, it can be a black box. We don't really know which are the dimensions that we're picking up and what our classification is based on. Maybe if we're not being very careful and we're not controlling our stimuli very well or our experimental settings very well, we can miss some confounding variables that co-vary with the dimension of interest, and that's what our SPM is really picking up. And it's really difficult, even by looking at the weights. People have tried looking at the weights or feature trimming, and I'm thinking about Federico, for example, the Martino. But I still think that is difficult to really pinpoint what are the information or the dimension, the stimulus that SBM is decoding. So can we explicitly model the effects of interest? And obviously we can do that using encoding on forward models. One very basic way to do this is just using a GLM analysis and build on the temporal history of your stimuli and the convolutional property of the HRF to see whether one cognitive model would be better than the other. So the way this works is, I'll just go very quickly over this for time. I'm already half an hour. This is just to show how this how we quantify the fit of the model. So cross by the R square. In an ideal world, our signal is equal to our design matrix multiplied to some beta weights that we optimized without any error. So we can derive this data, beta weights from one run and then multiply by the design matrix for the other run. And pretty much the distance between this model x2 by b1 and y2 will tell us how well the betas computed in run 1 will generalize to run 2, and then you can just compute your cross-validated coefficient of determination, which is the same as the normal coefficient of determination, safe from the sum of squared error. And here what you're subtracting is the f of x, which will be x2 times b1 minus the data points here. So in the 2015 current biology paper, again, the feedback feed-forward occluded region, we used this GLM encoding model to try and see where informative voxels were located. So we started from very simple conceptual model, whereby we reasoned that from a feed-forward point of view, the design matrix or the voxel that don't care about feedback information, would not really find any differences between these identical white occluders. However, if we did care, if that voxel did carry some contextual information, some fill-in information, these different occluders should elicit different responses. And so we can exploit, as I said, the temporal contribution of the event to test this with a very simple GLM. We can have our feed-forward model where we leave our labels condition unchanged. And then we have our feedback model where we randomly shuffle the position of these images randomly attributed to one condition or another. The idea being that the events explained by the model, if it doesn't care about this contextual information, should not be affected by this shuffling idea. And then we're going to compute the R-squared, provide R-squared for the two models for each voxel, and then through some Monte Carlo simulation, we can derive an empirical distribution and attribute a p-value, threshold it, and see which ones are the voxels that prefer one model to the other. And I said the cool thing of looking at this voxel model is that we can see where these informative voxels are located in the cortex. And so this is what we did here, a different threshold. We look at the percentage of informative voxels for all of our subject. And what we observe are results that are comparable to the decoding results, whereby the largest amount of informative voxel were located in the outer layers of V1. I'm going to skip this out because of time, but very quickly, just to say that there's a very difficult balance to strike in my mind when you're doing any type of computational model between the complexity of the model and the interpretability of the model. Adding more and more parameters will explain your model better. Yeah, sure, you risk overfitting it, but then it becomes difficult to interpret what these different parameters means. This is a paper by Michelle. I really like this paper, and I strongly suggest to read it, where she used competing models across layers with auditory stimulations in primary auditory cortex and non-primary auditory cortex. These models had a lot of parameters to estimate, but what she observed is that the two competing models were the spectral temporal modulation model and a frequency model. She observed that in primary visual cortex, this model only differed significantly in the outer layers, while if you move outside of the primary auditory cortex, then you got differences in deep, middle, and superficial layers. Read the paper, is really cool, especially if you care about modeling and auditory stimulations and auditory organization. So last part, I'm going to talk quickly about the population receptive field or PRF mapping. This work by Dumoulin, Serge Dumoulin, who kindly shared some of the slides with me. So what are population receptive field? Okay, so receptive field is probably one of the most widely acknowledged property of the neocortex, not only in visual cortex, but any cortex. And it basically tells us that single voxel respond to, in visual cortex, for example, in a different portion of the visual field. And they are selectively responding to visual portion of the field. When we talk about population receptive field, is because when we're in fMRI, within a voxel, we have thousands and thousands of neurons. So what we're studying is not the response property of a single voxel, but to a population of voxels, hence population receptive field. And the way this works is quite simple and elegant. They use a stimulus like this, some bars, gratings flickering at a given hertz and moving throughout the visual field, and record, in this case, bolt signal, but you can do this with any type of functional signal. And this is an example of a voxel from V2, while the participant is observing these moving bars. And then they fit a PRF model. So the simplest one that they developed in 2008, Dumoulin and Wandel, is just a simple 2D Gaussian model. There are much more complex models out there, but this is still a very relevant and good model that people still use. And so in this model, you have three parameters to optimize: X, Y, and sigma. X, Y would be the location of your receptive field, in position receptive field in your visual field, and your sigma would be the width of your Gaussian. And the idea is that if you, once the bar travels through the receptive field, you should see some sort of like activation within that specific voxel. So what you can do, you can then do a prediction of, and you can see how well your model is fitting your data. So you start from, it's a forward problem, so you keep optimizing the parameters until you find the best fit. Obviously, this is not a very good fit. You can change your X and Y to a different location, you get a better fit, and then you also optimize your standard deviation, and then you get somewhat of an optimal fit. So he performed PRF mapping across cortical depths. And what he observed is that the size of the PRF mapping changed as a function of depth. They were larger in inner and outer depths, and smaller in the middle depths. This is consistent with the idea of feed forward coming to the middle layers and feedback to outer and inner layers. And also, the very cool finding here is that it's very much consistent with the animal electrophysiology, where they observe comparable results. Last thing I'm going to show is how these, the attention field modulates the shape. Well, rather than the shape, I'm going to say the eccentricity of these receptive field. So they performed an experiment where they're asking participants to either pay attention to a specific portion of the stimulus, and they observed a shift in these in this PRF, in the central mass of this PRF. They have hypotheses of how this is happening, competing hypotheses. They are related to the gains in your synaptic clefts. But the cool thing that they did then is then go and look at the differences in this eccentricity difference across layers. And what they found is that the biggest differences in eccentricity was observed in the innermost depths, which is again consistent with animal electrophysiology of feedback exchange in inner depths. This is obviously anti-correlated with the gray-echo ball signal. This is gray-echo data, which peaks on the outer surface, as we've seen over the days. So this finding is even more, in my mind, reassuring, because it's not just following your univariate response sampling. All right, that's what I have, and I don't have a conclusion slide because I would just wanted to show you and make you aware of all these different models. I just want to make sure that I'm not putting the wrong message across. I still think that univariate amplitude is very, very important, and is a great way to look at your data and to analyze your layer effect, and I use it very, very often. But there are other ways, obviously, of trying to extract the relevant information from your data. And like Renzo was saying yesterday, you know, if you don't have the effect of interest before you despair, you can try and look at your data and plot your data in different ways, or try and extract whatever effect of interest in different ways. And with this, I'd like to thank you all for your attention, and my collaborators. Thank you.